Thank you. I thank the SRM University, particularly my good friend Professor Durgarao and the chair of the department, for other faculty members for this invitation. So every moment of it has been enjoyable. Because early on, I realized that I can ask good questions and pursue them methodically, doing experiments, utilizing all the techniques that are available, regardless of whether they're from chemistry or from physics or from biology, and apply those techniques and tools to my research. So from that perspective, we have learned both expected answers and also radically unexpected answers. So that kept me going on and on. And I must also acknowledge that my drive to sustain my career in molecular biology has been propelled by my students, postdoctoral fellows, and they have been immensely helpful in achieving whatever little that we were able to do. Very good question indeed. The way the balance is struck by cells is that there are editing mechanisms and also there are regulatory mechanisms. So DNA repair or recombination is not a foolproof or airtight process. It's also amenable for making mistakes. Like recombination as such is promoted by two independent mechanisms. We have what is called as a non-homologous recombination. As the term specifies, non-homologous recombination does not involve any homology and it is prone to making errors. Similarly, take DNA replication. As much as one thinks that DNA replication is an airtight process, it also makes mistakes. That's why you have all these repair mechanisms to iron out all the mistakes in the cells. Every day is excitement because every day when I talk to students, they come up with, or my postdocs, they come up with their findings. So we sit together, analyze the data, and it takes time even to come up with a tangible kind of possibility for observations what you have made. So sometimes we do make mistakes, but we make mistakes in interpretation of the data, not in the experimental aspect. See, interpretation of the data varies from time to time. But your experiments, what you have done, and data, what you have collected, have to be reproducible. So that is the beauty of science. The beauty of science is if your observation, collect some statistics, data with appropriate controls. See, controls in biology is very important. So that's why, you heard of a famous scientist by name Arthur Kornberg. Arthur Kornberg had this acronym, or rather metaphorical kind of a thing. In biology, what matters is controls and more controls. So controls will tell you whether your finding is wrong or right, because you have both a negative control and a positive control. So every experiment as a budding scientist, don't do an experiment unless you have a positive control and a negative control.